thanks for the introduction, and also thanks, of course, to the organizers for having us all here for a really exciting time. You might be a little confused with the title because actually when Jack first asked me if I wanted to talk here, then he gave me some title about like why we need the EMP, I guess. And then in writing an abstract, I figured I would put something that was maybe more close to some of the projects we're actually doing. So the title I have in the program is combining large and small scale studies, but the title that Jack sort of gave me was what microbial ecology has to gain from the EMP. So actually I've made a hybrid title that encompasses both of these, which is sort of what I'll talk about, is why the EMP is important. And of course we just had a really good overview of some of the really major questions that can be addressed. You'll hear a few of them come by again from me, maybe from a slightly different perspective. And also what I think some of the issues that we sort of have to keep in mind as we're trying to attack this, what we might call a grand challenge. So the stuff I'd like to discuss is first a brief, I would say very brief, sort of introduction to microbial diversity. And then some of the issues that we're just hearing about, why we should have it. And then the last part is also going into the points of why we really need to maybe think really big, but also not to forget to think about really small. So we talk about, and I talk mostly about soil-borne microbial diversity because I'm not a very wise person. I decided when we look at microbial diversity, I'll look in the worst place you could possibly, the most complicated, terrible environment to study. But yeah, we certainly wouldn't want to make it too easy for ourselves. And I think it's just the pure fascination with microbial diversity. If there's one place you want to be fascinated by it, the soil is a really good spot because obviously it's not just diversity per se, it's actually driving many of the ecosystem functions that are required of soils. And as we saw in the previous talks, the numbers of organisms is just so immense. And of course, you always have the analogies to how many things. And I always like I have a nice slide that actually does the Bill Bryson trick of actually writing out the numbers. And of course, you can see that if you compare numbers of stars you're talking about to the numbers of cells, it's just a larger number. It's not fair to put powers. But not only are there lots of numbers, but the diversity is, and that's what we're talking about here as well, is the actual species diversity is huge. Of course, we don't know how to define the species, and that's a whole different argument. But what really strikes me is that our estimates of diversity are totally flawed. So if I talk to colleagues who work with insects, and they're all excited because someone went to the tropics and found some new species, and they say, you know what, we might not even know half of the insect species on Earth. And I say, wow, that's amazing, but we don't even know a small fraction of the microbes on Earth. And in fact, we don't even know how many microbes there possibly might be in terms of different species. And the different estimates that can be made, from the millions to the billions or more. Of course, it depends on the definition. Well, that's just really striking. And actually, when we're talking about how much we know about the extent of microbial diversity on the planet, it was summed up very well by master's students in my course on microbial diversity at the Free University that I gave this past fall, where he said on his final exam, and he had to say how much microbial diversity was there. He said, and he wrote this down, and he wrote, it's basically like, wow, there are many more species than you can imagine. We got full marks for that answer. It's exactly correct, and that's about the level of resolution we have at present. Some people might articulate it differently, but that's pretty much where we're at. But also it's interesting, and this was brought up that we're at a very exciting point in history perhaps, is that we have an accelerated rate of diversity. If you look at Ernst Haeckel's Tree of Life, which he drew about 130 years ago, really a beautiful tree. Exactly, and some of the relationships are actually pretty okay. The whales or mammals too, all these kinds of things are on this tree. Of course, it has all of the microbes at the base of that tree, and the relationships of the major branches of life are totally wrong. But it's really nicely done. And it took us about 100 years to get to my high school textbook, which showed this tree of life, which is a lot uglier and probably has less, and it's equally and wildly wrong. And it just amazes me that within 30 years we've gone from this terrible understanding of the way life is organized on Earth to actually understanding the phylogenetic relationships of all these different organisms. And, of course, important to this group is the discovery that microbial diversity is so much broader than we ever would have thought of just 20 years ago, 30 years ago. And not only at the very fine scale of new species, but at the very large scale of new phyla. And this is continuing to increase as well. So to come, to follow up on the previous talk a little bit, so what are the major fields that I think now that will be governed by microbial metagenomes? Where do we find the microbial answers? And microbial ecology is really going to be at the heart of looking at major issues in the world. And I've listed a few of them here, and I'll give maybe just a couple words about each. Certainly the issues of climate change, energy, renewable energy, also human health and nutrition, industry and agriculture, and of course maybe most exciting to us is advances in ecology. So the idea of climate change, and I think this is Hurricane Katrina ready to hit New Orleans there. So if sea levels rise by just 1.5 meters, this is the way the Netherlands would look. Is that going to happen in 50 years from now? Well, this is my only political statement of the day. Well, probably it's not, because in Holland they can build these things like dikes, and they'll just build them higher, and it'll just be bigger, and it's not going to be worse. So technology will take care of that. But there are a lot of places like this, Bangladesh, that's not going to happen. So it's going to be a serious issue, and of course it requires a community approach to dealing with climate. But if we really want to understand climate change, we have to understand how climates change, not only based upon our influence, but also that of the engineers of the planet. And so if we talk about microbial engineering of Earth, we have to realize again that actually the majority of Earth's history has only known microorganisms. And what's really, I think, a good way to look at it is if we were looking at Earth from afar and we said, okay, is there life on Earth? Is there life on Mars? Well, you can see that there's life on Earth because our climate, our environment, has been shaped by life. So I think you just sort of turn it on its head. It's not that we live on Earth because it's a nice environment where organisms can live and get along. It's shaped to be that way, and that's why life is the way it is, because life has shaped Earth to be this way. And of course, if we want to understand climate change, we have to understand the principal drivers of climate change. Those are people, and until recently, those were mostly microorganisms. Of course, there's also plants and things like that. We can't forget about them. But clearly, that the microbial drivers of the planet have to be understood before we can understand how to deal with and understand what's happening with climate change issues. Also, in terms of energy, there's a number of things that are just going to be definitely micro-driven. And I'm convinced that most of our energy issues will be solved in some ways, or if they're going to be solved, they're going to be solved by microbes. So things like production of hydrogen gas, but also there are lots of different ways you can do this, and especially coupling this with light harvesting, biofuel cells, and people even think maybe doing this on the moon because you don't have things like to worry about your atmosphere. I don't know how you get the energy back to the Earth, but okay, that's another story. You can, in terms of all biofuel things, and how do you actually convert things from a particular biofuel to actually energy, artificial cells, which end up making what you want in terms of artificial cell factories. These are all things that are going to be driven by knowledge of microbial metagenomes. Also, when it comes to human health, not only will we now have our own genomes, perhaps, and then you'll know which things you might be allergic to or whatever, but also you'll definitely need to have your own microbiome so that you know exactly which medicines will work best for you and so forth. I think maybe some of these areas have promised a whole lot more than they might be able to deliver in the short term, but I think it's certainly an area that is ripe for funding at least. And of course, in the areas of industry and agriculture, I was used the idea that actually the majority of all bioproducts that we use, be it enzymes or antibiotics or whatever, do not come from some rare plant species in Brazil in a rainforest that is in the top canopy somewhere. It's just in the soil, and the fact that the majority of these compounds are in soil, but we only know a very, very small fraction of these obviously it follows that there's a wealth of biodiversity that's waiting to be unlocked for human use or abuse. And one point that's really important in this is that when we're looking for new products and things, we've really discovered now that we need to look in the right places. So, for instance, we have a good example of a number of colleagues work quite a lot on biocontrol issues. So strains which can, bacteria which can, for instance, impede pathogenic fungi. But we end up finding that most bacteria actually do that very poorly. But only when you actually have bacteria interacting with each other in a particular community structure do they actually have very, very large and important impacts. And actually then you can isolate those genes and those products which they actually make. So you really have to look at, you can't look at organisms individually in many cases. You have to look where they're actually interacting. And another area where clearly knowledge of the soil functioning is going to be more and more important is in the sustainable agricultural practices, where with the need for lower input types of agriculture, we're going to have to rely much more on microbial partnerships in agriculture. But maybe to this audience, the most sort of interesting and important things are what are maybe the ecological and evolutionary advances that we might expect or we might put as goals within EMP. So what are the basic scientific questions that we want to address? And we have already had a good list of them. I'll reiterate a couple of them from my own perspective. So what I started with is how many species are there? Well, I think that's a pretty basic kind of thing. It's really a basic question, and we don't really know. Or what's the total gene pool? And of course, how do ecosystems actually function? This is the most difficult one, perhaps. And also, from my own perspective, what are the patterns of this microbial diversity and function at different scales? And I'll talk about that now for a few minutes. So within these patterns of microbial diversity, I think I want to come back to the point that it's important to think big and think small. And maybe I'll go into why we need to do that. I would argue that to date, actually, we've mostly concentrated on scales of convenience. You can take a particular sample, you can examine it at a particular level. It's obviously difficult to sample all of Africa, but it's easy to sample one particular field in the Serengeti. The same goes for lots and lots of studies. At the same time, it's very difficult to study an individual microbe, whereas you can study a billion of them in your sample and destroy them all and then sequence a few of them. And this is really important, I think, because the drivers of actually what organizes the microbial community and the way organisms actually interact with each other and express all their activities is at a very fine scale. So the organisms only in their local area are actually interacting with each other. So this is, Jim Prosser had a nice analogy at, I think, the Soil Metagenomics conference, where he said, okay, so say we wanted to just pick 1,000 individuals on Earth, and we would like to understand how they all interact. Obviously, if we chose random individuals across Earth, we would never understand how they interact. But for instance, if we took, say, 5 people in a bakery and sampled those, and then took 5 people in a factory and sampled those, and 5 people on a football pitch and sampled those, then you would know which one was the captain, and which one was the defender, which one was the goalie, or which one was the baker, or which one was the clerk. And then you can figure out how these things interact. But of course, if we took one of each of these, we would know these things. So the drivers of diversity and interaction are local. But if we want to understand how they affect, for instance, the global carbon cycle, then obviously it doesn't matter what this individual microbe is doing. It matters what they're doing over a long period of time in a large area of space. So that's the difficulty we have, that both small and large are important. And I think I try to sum it up with this type of graph where the area in the middle is the part that we typically cover in microbial ecology studies, or what you might call is PhD student friendly. You can handle it in a short period of time, you can take some good samples, and you can examine it. But all the interesting stuff in terms of microbial interactions is all the way on the scale of microscale. But all the really interesting stuff on microbial impacts are maybe at a really large scale, and that's really difficult to handle. But with some of the things that have been developed recently in this EMP project, we can actually start